Hi everyone, it's Julia. We are going to talk about molecular technique, specifically blotting. Blotting is a technique for identifying biomolecules like DNA, mRNA, and protein at various phases of gene expression. It consists of the transfer of denatured fragments from the gel to a carrier membrane, which renders them accessible for examination with a probe or antibody. And these are the general principle of blotting. First is the electrophoretic separation of protein or of nucleic acid fragments in the sample, transfer to and immobilization on paper support, binding of analytical probe to target molecule on paper, and lastly, a visualization of bound probe. So these molecules in a sample are first separated by electrophoresis and then transferred on to an easily handled support medium or membrane. This immobilizes the protein or DNA fragments, which provides a faithful replica of the original separation and facilitates subsequent biochemical analysis. After being transferred to the support medium, the immobilized protein or nucleic acid fragment is localized by the use of probes such as antibodies or DNA that specifically bind to the molecule of interest. Finally, the position of the probe that is bound to the immobilized target molecule is visualized usually by autoradiography. There are different applications of blotting technique. One is the southern blotting. Its applications in molecular biology include the identification of one or more restriction fragments that contain a gene or other DNA sequence of interest, and in the detection of RFLPs used in construction of genomic maps. The second technique is the northern blotting. It allows in observing a particular gene's expression pattern between tissues, organs, developmental stages, environmental stress levels, and pathogen infection. The technique has been used to show overexpression of oncogenes and downregulation of tumor suppressor genes in cancerous cells when compared to normal tissue, as well as the gene expression in the rejection of transplanted organs. Lastly, the Western blot, alternatively known as immunoblot, which is used to detect specific proteins in a given sample of tissue homogenate or extract. The method originated from the laboratory of George Stark at Stanford. And these are the differences between southern, northern, and western blot.